Okay, 1 second. All right, we are live. Okay, cool. And I heard we had a question here. The aquarium in the back there is neocardinia or rainbow shrimp, and then there's actually one behind that, and that's a poison dart frog. Yeah. All righty, so let me share my screen here, and we will get rolling with announcements. Okay. Cool. Welcome everybody to the UWSP Student Chapter of the Wildlife Society's general meeting. Got a super cool presentation today with waterfowl. We have Jacob Sherba with us today. So without further ado, let's get started. So for dues this semester, we have a couple options for you, and those are $5 a semester or $8 a year. And there's a couple ways to pay. You can pay online through PayPal using my email. Otherwise, you can do a cash drop-off at my office hour from 10 to 2. If you're using the PayPal, make sure you use friends and family, and make sure to put your full name in the notes section for me so that I know who it's from.
All right, and to go out on projects this year, we need 3 things from you. We need a membership form, a hold harmless form, and dues to be turned in. I will put the link for the form in the chat here in a sec. Hold harmless has been sent out in an email from me previously, so just search your inbox for that. If you can't find it, please feel free to email me, and I will send you a copy of the form to fill out. We need all 3 items for you to be able to go on projects. I have a lot of people who've turned in just dues or just hold harmlesses, and we need both of those things along with the membership form for you to be able to go on projects. So as I said, I emailed out the hold harmless previously. You can fill it out using Adobe Reader, fill and sign, or you can print it out and send me a picture. Either way works for me. But when you do send it back, please put your full name in the document title and make the subject line of your email TWS hold harmless so that my inbox does not explode. Thanks.
Sweet. Thank you, Melinda. Lizzie.
Hello, folks. Tonight, our conservation topic is about wild horses and burros. I know many of you know this already, but just to clarify, the Wildlife Society is a national organization, and then we're a student chapter. So the National Wildlife Society made a call recently to reduce wild horse and burro populations. And the reason for this is because the population of wild horses and burros out west is just astronomical, which to the point where it's causing damage. Like the rangelands just can't support their populations anymore. And they're actually a non-native species. So recently they made an oral and a written testimony to the National Wild Horse and Burro Advisory Board. And what this board does is it kind of gives guidance on and like recommendations to the Bureau of Land Management, also known as the BLM, and also the US Forest Service. So the Wildlife Society called to reduce population levels as quickly as possible. Now, non-lethal like fertility methods have been being used so far, but it's just not working fast enough to the point where the populations just keep rising and nothing's really being done about it. So the BLM made a new plan to remove up to 20,000 individuals from the range each year and treat up to 9,000 of them with fertility control. So hopefully we'll start seeing some results here, and they're not calling for lethal action so much just because the outcry from the public would just be way too great if, you know, the BLM started shooting a bunch of horses and burros. So yeah, just kind of a current event, and also our conservation topic. Can move on. So our quiz bowl, we have 2 questions this time. The first is, what year was the Wild Horse and Free Roaming Burros Act signed into law?
Answers are rolling in.
Yep.
Few more folks. Give them like 5 seconds. All right.
Okay, you guys got it right. Good job. It was 1971. So yeah, a couple of them I threw in there to, I don't know, pretty out there. 2002, that's pretty recent, and 1857 is pretty long ago. But we can go on to our second question. About how many wild horses and burros are on U.S. public rangelands? And this estimate, just so you guys are aware, I took this estimate from 2019.
All righty. Couple more. 3, 2, 1. Ending it.
Oh, you guys were close. It's actually 88,000, and some of the more recent estimates have been even higher. Not quite yet over 100,000, but still much greater than what the rangelands can hold and also support like our native wildlife. So yeah, good stuff.
Awesome. Cool. Thank you, Lizzie. All right. So if you guys want to gain some more knowledge on wolves, or if you're just interested in their management in the state of Minnesota, we have a few webinars that the International Wolf Center is putting on. So the next one is going to be on October 19th, and that's Living with Wolves and Wolf Depredation in Minnesota. Should be a cool one. So if you want to register for that, there's the link. It's just wolf.org programs and then webinars. And that's a free webinar as well. So hopefully you guys register for that one. And then we also have the North Central section of the Wildlife Society has a bunch of talks coming up here pretty quick. First one on October 21st. And then on November 18th, we have our own Dr. Riddle having a talk on leading effective online meetings. So these should all be really cool. There's some really great folks talking, as well as our very own Dr. Riddle. And the deadline to register is October 15th for those as well. And this is completely free as well. So totally recommend doing that. Gain some knowledge from some pretty cool folks. Mackenzie.
Ooh, that's me. Hi, everyone. So this year's Midwest, or I guess it's next year, Midwest Fish and Wildlife Conference is February 1st through February 3rd of 2021. As of right now, it's virtual, and the abstracts have been extended to this Friday. So I have the link below. This is a really awesome way to present your research and network with professionals. And I will be sending out an application for being sponsored to go to this conference by our TWS chapter. So keep on the lookout for that. Again, this is a great way to present your research.
Awesome. Thank you, Mackenzie. Is Jacob here?
Yeah, I am. Can you guys hear me?
Cool. Yeah.
Yeah, so I'm finishing up trapping. I got about a week or two left before it gets too cold to do. But basically I'm doing a presence absence at 2 different sites in Schmeekley. One of the sites is our burn site, Burrard Oaks, and then the other one is along the Trail of Reflection. So if anyone wants to do some small mammal trapping, we've caught a lot of white-footed deer mice, and then we're starting to get a lot more flying squirrels, and then there's a little bit of diversity of catching a couple of shrews. So if anyone's interested, just email me right below there.
Awesome. Thank you, Jacob.
Hi everyone. So CNR Bible study is tomorrow at 7 to 8, and it's on Zoom. So the link is right there on the slide. And we haven't been getting too many people out, so we'd love to have more people. And if you have any questions, you can email me, or you can email Megan on the emails on the slide there.
Awesome. Thank you, Melina. Muscle project.
All right, everybody. We are actually able to get out into the field. We're actually in the field now, as you can see. We just brought in a couple mussels, and this is probably the biggest one we have for the day. We're looking to go out this week. Wednesday and Thursday is probably the nicest days. Weather depending, we'll keep going. You can email either Isabel or I. Our emails should be in that presentation. If not, let me know somehow. Times are there. You will have to check out waiters from the stock room, and we will let you know the time. Just meet outside of the west side of the TNR, and we'll get a caravan going to get everyone there. Yeah, just send us an email and we'll get everyone out. We are looking between 4 to 8 people to get us out and going. Numbers will depend on how many of us co-leaders can make it each day. Are there any questions?
Cool. If not, thanks, Luke. Deer project.
All right, hey guys. So the Deer Project is looking for 1 or 2 co-leaders this year to join us, and like everything else, freshmen and sophomores are highly encouraged to apply, just because the only way to get your experience is to just get out there and do it. And like, just for like a short summary of what we do, we trap during the winter months, like below 32 degrees, so we don't overstress the deer. And we collar and ear tag adult does, and we also ear tag bucks and fawns if we catch them as well. Typically we bait traps Thursday nights and then check traps early Friday morning. And Nate will send out an application after the meeting tonight, and any questions and the applications, please send to me, and my email is linked below. Thank you.
Awesome. Thank you, Jeffrey. Gray Squirrel.
All right, hey guys. So Gray Squirrel Project is also looking for co-leaders. We're a spring project, and like Jeffrey also said, highly encouraging freshmen and sophomores to apply. It's definitely, you get a lot of hands-on animal handling experience. I know Nate sent out the applications, I think early this morning, so you can search for that in your inbox. You can just send me the applications. My email's up there. And if you have any questions, you can let me know or any of our other co-leaders. So.
We're also looking for like 3 to 4 co-leaders, because I'm going to be graduating, and then it'll be just Maddie and Rebecca. So we're going to be looking for quite a few. So.
Yep.
Cool. Thank you so much, guys. Bear project.
All right, folks, there's just 2 days left to submit your applications. They're due the 15th at midnight. We're still, for everyone who's applied already, thank you. We will let you know after the 15th if you have gotten it or not. So for anyone else who's yet to send me an application, like, my email's there, and you can direct any questions to me as well. Thanks, guys.
Cool. Thank you, Bear project. Saw wet.
All right, guys. We are looking for preferably freshmen for the Sawwhat Owl project. We need probably 2, so we will be going through a lot of applications as well. But this takes place in Schmeekly this year, not at Sandhill, and we caught a few of our first owls this weekend. So we would like for you guys to be able to come out. If you're applying to be a co-leader, we'd like you guys to come out with us before the season ends. So if you guys have any questions, I have my email at the bottom, and the applications were sent out, I think, earlier today, right before the meeting announcement was made.
Cool. Thank you, Carter, from the Sawwhat Owl project. Yeah, guys, so tons of opportunities to get involved. A lot of projects looking for co-leaders, and I'm sure more will pop up as the semester goes on. So don't be afraid. Get out there, apply. The worst that can happen is it's a no. So you'd be surprised. All right, small mammal.
Hey guys, the Small Mammal Project is trapping out in Schmeekley right now. We check our traps at 7 a.m. and 7 p.m. and we meet at the Schmeekley Visitor Center parking lot. Check your email for the sign up, or I can send the link in the chat. We'll also be looking for some co-leaders soon, so look for the application for that. If you have any questions, feel free to email any of the co-leaders up there on the slide.
Awesome. Thank you, Shelby.
All right, that's me. So last week I introduced this volunteer opportunity and we did get some interest in it. So the October 26th date is full to volunteer at the Primate Sanctuary in Westfield, Wisconsin. After getting a little more info, it'll be mostly helping build, I think she said it was some enclosures as well as some winter, not renovations, but updates. And so if you guys are interested, it would be the 27th because the 26th is filled and we have room for about 3 people because I might be going and driving for that. If you have any questions, email me. My email's on the slide, as well as Sarah. Her email is right below mine. She's a volunteer there, and she has a little bit more information than I do. But it looks like it'll be a fun time. If you have any questions, email me. Otherwise, look at their website. It's really cute.
Awesome. Thank you so much, Danny. Seems like a pretty cool opportunity. Nathan.
Hi guys, so welcome to Subway Club. That's where I am now. You know, Pluton Cahoot in college. So starting tomorrow, we're gonna have our food. If you want, you can start racking up points. And at the end of this year, I'll keep track of every about the whole time, and then we're gonna dole out some to those cool ones who really time to get those points. Nate, you can hit it. All right, tomorrow's gonna be Bahoot. Really thought about that time. Link. I can throw it in chat too. Also should be in the student announcement tomorrow. It'll be at 6:30. Welcome. It'd be a good chance to meet some people that are officers might be there. So get in contact with them. There's a, but it'd be a great have.
Alrighty. Thank you, Nathan. Shelby.
Hey guys, so every Friday we feature a photo taken by one of our members. So if you've taken any wildlife photos recently or in the past, send them in to be featured on Fauna Friday by DMing our Instagram page, or you can email me. And then you can follow our student chapter on multiple platforms. You can like us on Facebook, follow us on Instagram, and you can add us on Snapchat through the code up on the page. If you know of anyone who couldn't make it to this meeting, or if you can't make it to a future meeting, our meetings are recorded. You can watch them on our Facebook page or our YouTube channel.
All right, awesome. Thank you, Shelby. Cool. And then, so quick announcement, everybody. CNR scholarships are live, and I believe the deadline is later this week. So get out there, do those scholarships. There's a ton of money available, all for CNR students. So go check those out. I believe you got an email for it. And I think I'll send out an email with the link to those as well later in the week, just so you have it. So next week, we have Janelle Marie, who's an airport biologist with the USDA Wildlife Services in Hawaii, talking about her journey and how she ended up where she ended up. So should be a super cool talk. But this week, we have Jacob Sherba with us. So without further ado, I'll stop my share and give him his time.
Thank you, Nate. Really excited to be back with Stevens Point TWS. Thank you guys so much for having me. I'm gonna see if I can't get this started. Okay, can I just get a thumbs up to make sure that you can see my screen and that you see the presentations? Perfect. Okay, so as you can see right off the bat, I've very tactfully named my presentation, Where the Hell Are All the Ducks? If you are not in the waterfowl field, or if you are just kind of breaking in, this is probably one of the biggest questions you will constantly be asked by waterfowl hunters at any given point. Usually it will follow up with some story about the cool birds that they have had a chance to shoot or work with. But where the hell are all the ducks is probably one of the most common questions we tend to get just from the average hunter out in the field with us. So before we jump into the ducks themselves, let me give you a little bit of information about who I am. I am a Stevens Point grad. I graduated from the wildlife department in 2018. And then just to set the standard here, I am team bird, never I actually can't say I never stepped foot into mammals. My advisor was Shelley Dubé, so I inevitably stepped foot into mammals at some point during my my time at Stevens Point. But currently I'm a graduate student at Clemson University working on my master's and studying waterfowl population and disease ecology, specifically working with wood ducks in the southeastern United States. So while I'm currently working as a waterfowl ecologist, my background and training is almost entirely in wildlife disease ecology. I sort of fell into waterfowl ecology after working on a wood duck project with Dr. Jake Straub, who was our previous Kennedy Center chair before Dr. Sedinger came. So disease ecology work at Stevens Point focused almost entirely on wildlife parasitology. These are photos that I took while I was working at Point. I was trained by my dear friend and colleague, Dr. Shelley Dubé, who I think is haunting around this Zoom call somewhere. I swear I saw her name come up. And probably my favorite project while in Point was investigation of gapeworm parasites in greater prairie chickens. And that's this beautiful parasite down here in the far right corner. Additionally, so this project is, I spent almost my entire 3.5 years at Stevens Point working on this project with Dr. Dubé, and it's currently in review with the Journal of Parasitology right now. Other projects I worked on included just a general parasite survey of greater prairie chickens. I assisted Dr. Dubé with a study on the effects of black flies on sandhill and whooping cranes. And then finally, I spent some time working as a curator's assistant in the Parasitology Museum on the fourth floor of the TNR. Oops. So following graduation in 2018, I was hired by Clemson University and a nonprofit organization called Nemours Wildlife Foundation in coastal South Carolina, working as a field technician investigating wood duck recruitment in the Atlantic Flyway. So we were there to collect data on how nest box and habitat characteristics might influence box selection by wood ducks and how this in turn might affect their recruitment. The pilot study went very well because after it concluded, I was invited on as a graduate student at Clemson by the PI to not only continue the recruitment work in Georgia and Florida, but to bring in a disease ecology chapter to my thesis as well. I snuck it in there. Now, most of you, I'm sure, are familiar with wood ducks. They're a very popular species across both of our flyways. But in case you aren't, I want to cover just some really basic species biology just to kind of get us started. So they are a very heavily harvested species, usually the second or third most popular and most found species in hunters' bags. They were popular, as far back as we know, but they were too popular for a while because the species was nearly extirpated by the early 20th century in the Atlantic Flyway, due primarily to overhunting, as well as a loss of important forested upland and wetland habitats. They are cavity nesters, secondary cavity nesters, so with the loss of their habitat, they lost a lot of those cavities that they would have used to nest in. They will also utilize artificial nesting structures, though, such as these wood duck boxes you see in the photos. And it's it is thought that these nest boxes are partially responsible for the revival of the species as a whole. They are also highly phylopatric, with around 70% returning to the same site to nest and 40% returning to the very same box that they had previously nested in to continue nesting. Now, with, I believe Dr. Sedinger teaches it now. If you've taken PopDi with Dr. Sedinger, you would probably already heard the term recruitment before. If you haven't though, recruitment essentially refers to young being added to the fall population through reproduction by adults in the spring population. So the last time a recruitment rate for wood ducks in the Southeast was done was in around the 1980s, late 1980s by Dr. Gary Hepp and some other colleagues. Dr. Hepp is quite literally a wood duck god. He helped write so many just seminal papers on the species as a whole. But he and his colleagues calculated a recruitment rate of about 5.2%, which is really low for the species in this area. We want to see that number at about 10 to maybe even as high as 12%. Now, in 2019 and 2020, Hepp and his colleagues reanalyzed their data with some added data, since they've been going essentially since the 1980s on this stuff. And they determined that box nesting populations of wood ducks are declining by roughly about 19% per year. And additionally, they determined that box nesting populations are insufficient to sustain the population as a whole without immigration of hens from natural cavities. So this is a slide I throw in most of my presentations, but this is just something so you guys can see how huge this study is. Just to give you a time frame, this study is set to extend for another 6 years, give or take. And these are all of the collaborators currently involved in the study, with more expressing a lot of sincere interest. So we have collaborators as far north as Delaware and Maryland, and as far west as Mississippi State and LSU. So a lot of really great state, federal academic institutions and agencies being involved in this study. So what all is happening right now then? We have finished a very successful pilot study in 2019. So now in 2020, there are 3 graduate students currently working on this study, in addition to the many state and federal staff, as you saw in the previous slide. So graduate students are collecting data in Georgia, Florida, North Carolina, South Carolina, and Louisiana. And because we're all doing a very similar project in the form of collecting data towards estimating a recruitment rate, as well as looking at a rate of return for yearling hens and adult hens, we were tasked with coming up with additional projects that we could use to make our respective theses stand apart from each other. So other projects that are ongoing at these sites include investigating adaptive habitat management strategies to decrease predation and increase recruitment, monitoring box usage by both wood ducks and black-bellied whistling ducks, and to see if there's any overlap or competition between the 2 species for the boxes. And then for me, I'm looking at the microbial ecology or the microbial environment of these nest boxes to see how these microbes may potentially affect nest success and inevitably recruitment. Additionally, all sites are investigating long-term. This is one of our long-term goals, I guess I should start with. One of our long-term goals for the end of this study is inevitably to determine whether or not it's even worth continuing using these artificial nesting structures on the landscape. Because especially now that we have gotten much better with our forestry practices, these natural cavities are not as in demand as they once were. So is it even worth using these nest boxes? Because HEP's data certainly shows that maybe they're not worth keeping right now. That was only one site though, so that's one of our long-term goals for the study as a whole. So first, just a big collective we've got babies. This is a huge part of our study, is actually capturing the wood duck babies and marking them in a way that we can keep an eye on them and see how many of them return in the following years. So my primary field sites are in Florida and Georgia. To mark the babies though, we had to be clever because we couldn't use a band. If we used a normal size band that we would use for the hens, it would slip right off the leg. And we can't use a smaller band because that would constrict growth. And we don't want to see one-legged ducklings running around. So if a couple of you just in the chat, just from what you know of waterfowl ecology and just management as a whole, what are some options that maybe we could use to mark ducklings? I'll give you a couple seconds. Yeah, web tagging was the first two things that popped up. And yeah, that's exactly what we use is web tagging. This is our sole method. We pitched around some other ideas. LSU actually pit tags their ducklings. They apply a pit tag between the shoulder blades. And they're doing a cool side project right now where they're looking at web tag retention and if it's maybe makes more sense to pit tag over web tag, just from a tag return and retention standpoint. So, oops, that is not where I want to be. Oh yeah, it is. Okay, so this method of web tagging allows us to mark the babies in a way that they will retain the tag, since obviously we can't use the bands on them. So this is a pretty traditional mark recapture method. It's something that's been used since the late 1980s, I think, is when the paper came out that described this process. And essentially the way it works is you make two small incisions in the webbing of the foot. And the pictures going from left to right kind of detail the process for tagging the babies. But you make two small incisions, this tag slides through the holes, and then you seal it from the top. And the top is where the number is located, and the bottom, you can see on that far right picture, it says CU for Clemson University. That was from our pilot study. Now that the study has expanded into so many states, we now have the state initials on each respective site's web tags. So this way, if hunters find, if they harvest a bird like this, they can report these tags just like you would any other USGS lag band on the reportbands.gov site. But do they work? You know, it's a method that a lot of people use. There hasn't been a whole lot of data on tag retention. So do they work? Oh, and the other thing I want to point out too is that little equation down in the bottom left-hand side of the screen is what we use to calculate the rate of return for wood ducks. So we take the total returns of the web tag babies divided by the total tagged from that field season that we tag them in, subtract the total dead, and then multiply it by 0.5, which is a sex ratio, 50-50 sex ratio. This is pretty well detailed in the literature and actually can be seen pretty good in our data too. Most clutches are about 50-50 male-female ratios. And that number will provide us with a rate of return for wood ducks. So then kind of circling back to that question, do web tags work? Yeah, they do. This is a photo from this year. My colleague who works in South Carolina recaptured this yearling hen who is now an adult, and we were able to throw an adult leg band on her. But those are the web tags from our 2019 pilot study. So 12 yearling hens returned to South Carolina this year, gave us a rough rate of return of, I think it was like 5% or so. But out of those 12 birds, 10 retained both web tags, and the other 2 birds kept one of them. So we would consider that a really huge success and a perfectly fine reason to continue using web tags at our sites. So then on the topic of microbial diversity on wood duck nesting structures. So this is more my cup of tea. I am getting pushed further into population ecology, but my background is in disease ecology. So this is the stuff that's really interesting to me. So we have sterile swabs that we will use on the eggs in the boxes. So we will take randomly select 3 eggs from the boxes once incubation has started. And if you are not someone who's worked with waterfowl or with wood ducks in particular, we generally know incubation has begun in a box when there is a mound of down on top of the eggs. And that's usually our clue as to when mom is starting to incubate her babies. So we randomly select 3 eggs from the boxes, swab them with the sterile Q-tip, and then float those in a couple milliliters of sterile buffered saline. Those go back to our microbiology lab at Clemson, where they sit in a -80 Celsius freezer until I'm able to actually get my hands on them in the fall semester to plate them up. We use 2 different types of growth media to take into account the types of bacteria that we might be growing. And then each media is grown or is incubated at different temperatures to take into account just ambient temperature as well as roughly internal body temperature. And so these methods in particular provide us with the potential to grow some really cool stuff. So the once the plates are incubated, we check them at 24, 48 hours. They're done incubating at 48 hours. We check them at 24 though to make sure there isn't just like a lawn of fungi grown across the plate. If that's the case, we have to try and regrow the bacteria again and maybe take it out of the incubator a little earlier than we were expecting. But after 48 hours, we remove the plate, and those 4 little bacterial colonies that I have circled in red, that's what we're looking for. We want to see bacterial isolates like that so that we can remove those isolates, replate them, and try their species. So we're looking for similarities between the boxes. There isn't a whole lot of literature about what types of bacteria are found in these boxes and what sort of effects that they may have on the wood duck eggs. So that's one other thing that we're working on right now too, is finding some money to run some 16S rRNA gene sequencing with the DNA or with the bacteria as well, to where we can actually try and narrow that down a little bit further to see what we're looking at within these boxes. One additional aspect of the study that's I think is really cool too that we're doing is we are using a couple of different types of shavings within these boxes. So we're trying 2 types. So we're using aspen shavings and cedar, both of which are pretty commonly used by managers across the United States or across flyways who have wood ducks. The cool thing about cedar shavings though is they have a lot of natural oils that promote antimicrobial growth, while aspen does not. So we're keeping track of what boxes we took samples from and what sort of shavings they have in there to see if there's any differences between the microbial communities grown within those boxes. So hopefully I'll maybe be able to come back next year and have some cool microbe pictures for you and some kind of cool results for what we found from the boxes. So just to kind of reiterate the title of the presentation in terms of where the hell are all the ducks, where the hell are all the wood ducks? Honestly, the short answer is we don't know. At least in the Southeast, we don't know. Populations, as you saw, are declining due to these, box nesting populations are declining. They're not adding to the population as a whole. And you talk to hunters down here and they'll say, yeah, I've barely seen any wood ducks at all in the last couple of years. So for the Atlantic flyway, in our case, estimating recruitment properly to be able to provide an updated estimate for recruitment, as well as some insight into populations of wood ducks in the Southeast, is probably going to take another 3 to 5 years of data collection and analyses. There are my colleagues and myself will graduate in May 2022. There will likely be another round of master students to go out and collect more field data that we are collecting here now. And then they will be hiring a PhD student at the end of it all, who has the super fun job of actually calculating the percentage for recruitment. In terms of management considerations that come from this project, if you publish anything with Journal of Wildlife Management or write anything for TWS, generally wildlife biologists like to hear what management considerations are for our project. What are our end results for management? So the biggest one is going to be establishing more information on populations of wood ducks in the Southeast. This is a really huge thing. They're a popular duck. People want to know where they are. Additionally, we can whether or not nest boxes are useful and whether or not they should be kept on the landscape. Habitat management strategies to abate predation from the boxes. Snakes, they find their way into these boxes. You can have a box with absolutely no trees around them, the perfect predator guard, and somehow you'll magically find a snake in the box. So trying to figure out some habitat management strategies to abate predation. And then finally, what sorts of shavings managers maybe could or should be using in the boxes to prevent infection and nest failure due to microbes. So thank you so much again, Nate. That's really great. Like I said, being back with my TWS group, and it's so awesome to see so many people still coming to the meetings, even over Zoom during a pandemic. So great job, guys. Keep it up. This is a really important part of the education. So my information is down at the bottom of the screen. Honestly, do please feel free to reach out to me if you have any questions that we may not get to tonight. But at this point, I'm open for questions. I don't know how you guys do it, if it's in the chat or if you just turn on your video, but I'll open it up to you, you guys.
Thank you so much, Jacob. That was an awesome presentation. Yeah, guys, if you want to either type your questions or unmute yourselves and ask, I'll end the live here and we'll have a little discussion.